First of all, thanks to ARM for giving me this opportunity. It's a challenge, as you all can agree to in this room, to develop a regenerative medicine treatment, but it's probably even more challenging to keep you awake now this late afternoon, the last day of the conference. But I will try. So I hope you are bored of listening to cell therapies and gene therapies, because now we will move over to the third leg of regenerative medicine, being tissue engineering. So we are now going into much larger drugs than we have heard about previously. Namely, I will talk to you about organs. So my name is Petter. I come from IKEA land, Sweden. And I will tell you about our story. So I'm starting a little bit from behind. I think you can imagine how fantastic it is to have, for 8 years, been working in all kinds of preclinical model the day you are treating your first patient in your first clinical trial. And that happened more or less exactly 1 year ago, and that was here in Spain. I will come back to that. So Verigraft, we have a history from university, we have a history from Karolinska Institute in Stockholm. Today we are, however, located in Gothenburg, which is on the other side of Sweden. So we are privately owned from all over the world, and I think we have a very strong but small company. So talking about organs and talking about transplantation today, there is a challenge when it comes to transplantation, and that is when you transplant something from one donor to a patient, you will have an immune reaction. That is possible to handle with all the immunosuppressive agents we can use today, but that comes with a price. So that price is coming because the immunosuppressive agents are one of the most heavy or drug group of drugs connected with heavy side effects. But before diving into that, I will give you a little personal reflection about this field when it comes to organs and organ transplantation. So this is a lobster, as you can see, and I'm not here to talk to you about catching lobster or eating lobster. I have a lobster typically in my presentation to show you that nature is full of regenerative medicine capacity. You may know that if you break off a claw from a lobster, there will be a new claw growing out within only a few months. However, if you break off an arm or take your chainsaw and release an arm from a human being, there will be no arm growing outright. So our bodies have a much less efficient regenerative medicine capacity than some of these animals. So therefore, can we create organs? And sometimes you see these type of illustrations in literature, and I mean, I can hear people saying that, wow, now they can get deaf people to hear again. But I think you can imagine that this is not making anyone hearing again. This is a simple chondrocyte formation that is not really science fiction. But this one is really science fiction. This is actually taken from a company doing bioprinters or 3D printers. And is that a reality? Can we bioprint or 3D print a heart today? The answer is a clear no. We cannot print a big organ with all its function and get them clinically ready today. I'm a strong believer in this. I think we have to wait some 20, even 30 years before we do those type of organs at a scale where we can start to help patients. However, what we can do today is not really to create organs, but we can make organs ready for transplantation and much better when it comes to transplantation. And I will tell you why. And back again to the problems. When we transplant, we cause an immune reaction. So that can be handled with all these drugs, but that is a lifelong treatment. So it's a high cost. Therefore, of course, we transplant if we have a severe heart failure. We need a new heart, otherwise we die. But for many of the less vital organs, like the one my company is working with, vascular grafts, nerves, heart valves, We can live without them, but with a much lower quality of life. We are not transplanting them. So at the end, as a consequence of this, we do transplant large organs, but not the smaller organs. But does it have to be this way? I think no. So is there a solution to this, and how can we do? So we think we can do it without use of any immunosuppressant and still no rejection. So a little bit short about the technology. So this is a donor donating a blood vessel. What makes the blood vessel having an identity is mostly cells and DNA. So if we can strip this blood vessel from cells and DNA, we will have something that more or less lacks identity. We call it a scaffold. It has more or less exactly the same three-dimensional structure when you take off the cells and the DNA, like this. Remember that these structures consist of proteins. They're very easy to freeze and store for a long time. But we need another process. We need a personalization process. And this is something that people have worked with for decades. So typically when we personalize tissues like this, we use pure population of cells. You can, for example, take a syringe and take out bone marrow from your hip. You need to identify the cells you are looking for in the bone marrow. You need to isolate them, you need to expand them to a certain number. These are processes taking typically 2 to 4 months, up to 6 months, involving a lot of cost and a lot of regulatory hurdles when you culture cells for a long time in vitro. So we have developed something that is completely different. So as you see here, we are sampling not pure population of cells, we are sampling blood. With this blood, to this blood, we add some enhancing factors, we call them, something that is speeding up and facilitating the processes. So together, the scaffold coming from the donor and the blood solution coming from the patient, we mix them in a very simple bioreactor. It's simply a peristaltic pump that is facilitating this for a while. We can make this process working in less than 10 days. So instead of many months, we do this in less than 10 days. And we think that is, of course, comes with a lot of advantages like lower cost, less time consuming, and less regulatory questions, because we are not handling any cells directly. We are not culturing or expanding any cells. So in less than 10 days, from our frozen scaffold and blood from the patient, we are ready to produce, or we are ready to transplant this to a patient. So does this, we believe this is today the most biological way of replacing a blood vessel. So does this work? So I'm not having a lot of preclinical data with me, but this is from a porcine model, 1 year in vivo, where this graft has been, in this case a vein, has been introduced to the porcine model for 1 year. On this photo you see angiography, where you see that the blood is nicely flowing through the graft, and on this nice picture you see the graft being covered with endothelial cells. So it looked like it worked well. So now to the clinical journey. I told you that we are now 1 year into our first clinical trial. Where did we start? I mean, our bodies are full of blood vessels, so we decided to start in the groin region. In the groin, we have arteries leading the blood down to the feet, and when we walk with the muscle pump, the oxygen-free blood is going back to the heart. Unfortunately, since we are sort of animals on 2 legs, the blood tries to go down by gravity to the feet again. However, let's see, Mother Nature has developed a very simple but very efficient system in the groin, where we have one or a few valves, pair of valves, that are simply opening from when the blood is coming from the feet up, and when it tries by gravity to go down, they are closing again. So very simple and very elegant. However, for more than 1 million patients in Europe and the US are suffering from failing valves. When you suffer from that, you are in a state of chronic venous insufficiency. That is the name of the disease. Starting then, the blood is not controlled, it's going down to the feet. The patients are experiencing swelling of the leg, a lot of pain, but unfortunately, and gradually, as this disease, chronic venous insufficiency, progresses, open bleeding leg ulcers are formed in the more severe cases or the forms of the disease. So this is something that is not treated today. Of course, we are taking care of the symptoms, so we are giving them painkillers, we are taking care of the wounds, but there is no cure for these patients. And again, the reason is that we tend to do not transplant because the patients then have to live the rest of their lives on heavy immunosuppressive agents, increased risk of cancer, etc. So instead, we think that now we will do this. This is our product. It's simply a 5 to 6 centimeter long piece of the vein carrying these valves that we are surgically introducing to the patient's vasculature. And this is real life. I don't think you can hear it here, but it's actually our laboratory model where we are pumping a solution from the bottom up here, and I think you can see that the valves are opening and closing. And this is done for 500,000 cycles in the labs without any problems with the valves. So it seems like working. So we have now this trial ongoing. It's a small phase 1-2 trial, 15 patients. And the goal is, of course, to go to the market in Europe and as quick as possible and try to help as many patients as possible. So we are talking to the regulators in Europe, we are talking to FDA, and we have just initiated some activities to also go clinical in the US. So our business model is simply to be a manufacturer of these type of grafts and provide to the healthcare systems. At least in Europe and America from our own facilities, then we will out-license the model. And we have, of course, a lot of support for the business model. It's a strong reimbursement and health economical analysis behind this. So a typical patient with this disease are having a dramatic decreased quality of life, early retirements, on sick leave, etc., cannot go to work. So this is our lead product. It's called PTEV, Personalized Tissue Engineered Vein. We do have follow-on projects and product candidates, so we will go into the field of personalized arteries. That is a bigger field or bigger market with a lot of big companies already having an established market. That is mainly synthetic grafts. So synthetic grafts are on that market. We also work with nerves, urethras, heart valves, etc. So this is sort of a platform technology. I would like to end up by saying something quite exciting about the future. Today we are using, as you heard, donated human material. But you can ask me the question whether this will be a limitation, and it may be. So we think there are other donors out there, and those donors can be animals, so xenogeneic material. We have now our second product, most likely based on bovine material. So the cow is the donor. We bring it in, we take off all the bovine cells and DNA, we personalize with human material. Maybe a little more regulatory tricky, but still, it's definitely part of the future. We are not stopping there because I know that in this conference we have many believers of a third technology being printing and bioprinting and 3D printing, and we think this is a simple protein structure. So in the future we will be able to bioprint these structures and provide like our product development. So this is just a project where we are using the expertise of bioprinting. We should not talk more about that. Sweden is a good country right now because we are investing a lot of money and resources into this field. So CCRM Nordic on the bottom left is the new thing here. It's an expansion of the CCRM in Toronto. So that is established now in Sweden. We have a lot of EU money behind us besides the money from the investors. So my last 20 seconds is just to tell you that we are fundraising now, and should you think, anyone in this room think it's interesting, there are a lot of arguments for why there is a good thing to invest in a company like us at the moment, I believe. So that is very good. So let's stop this and let's end up with my little lobster with one big claw and one small claw. But as you see from this lobster, and I hope you can agree that this is a fantastic opportunity. So let's regenerate the future together. Thank you very much.